and welcome. Today we're kicking off the Carey Institute Fall Scientific Seminars series. Thank you all for joining us, both in person and virtually. For those joining us virtually, just a reminder, if you have any questions, please put them in the chat and we will do our best to answer them during the Q&A section. For those joining in person, Dr. Beverly will be joining us for lunch in the lunchroom, as well as for a happy hour at 4 p.m. today. And with that, it is my great pleasure to introduce Dr. Daniel Beverly. He's joining us from the University of Indiana, Bloomington, as a postdoctoral research fellow. He did his PhD at the University of Wyoming, which is actually where he and I met. He was a PhD student looking for field technicians to help with his PhD thesis, and I was a fourth-year undergraduate anthropology major skipping classes to read about climate change. So he gave me my first environmental job, which involved snowshoeing up mountains in the snowy range, carrying solar panels and car batteries and shotguns, all to measure winter water potentials in western coniferous forests. And if it sounds like tough work, it's because it was, but I loved it so much I became a forest ecologist, so I actually have Dr. Beverly to thank for that. Dr. Beverly's research is in plant ecophysiology and in hydrology. And something that I really like about it is that he asks questions around plant and ecosystem productivity across systems and scales. He's worked in western coniferous forests, as well as eastern deciduous forests and tropical forests as well. He's looked at deserts and agricultural systems, and he's asked questions from scales all the way from the tiny individual leaf to whole plant scales, and then all the way up to the entire ecosystem. I've also always really admired Dr. Beverly's curiosity, his generosity, and his collaborative nature, which I think really shows up when you get to hear about his research. So with that, I will turn it over to Dr. Beverly.
Thank you, Jazz, and thank you guys for all being here. I'm super excited to actually come and visit you guys and see the Carey Institute, and as well see your amazing campus, and also share the work that we've been doing for the last few years at Indiana University. So just to set the scene, I work primarily in eastern deciduous forests. So this is our Flux Tower site. So this is the Morgan Monroe State Flux Tower, which is in southern Indiana. But this tower site's been running for 25 years, and it's capturing a long-term flux and energy dynamics across this unmanaged ecosystem. And just before I begin, I just want to make sure I acknowledge my funding agencies, my collaborators, who all have helped me through this journey, especially Ameriflux and the O'Neill Skill at IU. So when we think about our motivation for doing this research and thinking about the questions that I think, this is one of the primary figures I always come to. When we think about this over time, we can see about the CO2 fluxes. So we have the top panel representing the CO2 emissions being produced, and we see this nice linear increase around 1950. And then when we look at the below side, we can think about the fate of these different pools. And we can primarily break this up into three major components. We think about the ocean pool, where the oceans are absorbing a bunch of the CO2 being emitted to the atmosphere. Alternatively, a bunch of the CO2 is being emitted into the atmosphere and then just staying in the atmosphere. And so we're getting a rise of CO2 concentrations that we all are accustomed to seeing. And the other major sink from this figure really represents the terrestrial land sink. And we can see that there's a huge amount of interannual variability, and we want to understand the dynamics driving this. There is also a new sink that is being really pushed by the US government. And so when we think about this, we think about CO2 capture systems. And there's been a huge investment on how can we add a new layer or a new slice to this cake. And so we're investing billions and billions of dollars in tax revenue into this. But we already have a really amazing source. And we think about the forests and how we manage them. We're already getting a 25% reduction in our CO2 emissions just from the forest alone. And it would be very valuable if we could actually tease this apart a little bit better. Can we harness the power of the forest? Can we harness the power of the trees? So today, I kind of want to talk about some of the options and some of the limitations that we're going to be doing as we progress through this over time. Harnessing this power is a much more complicated problem because of the spatial heterogeneity. If we think about this at a global scale, we can look at this figure looking at the globe and the CO2 source and sink dynamics across spatial domains. And we get some really awesome hotspots in the tropical forests. Also in the eastern forests of the US, India and China also are really big hotspots. But we also have these really, really void zones, especially in the US, the deserts, and in the southwestern US. And this is also accompanied by another problem when we think about this on a spatial dynamic. So we have this spaghetti plot. We are pretty common to seeing this, but we can look at trying to predict the annual land flux. So we have time on the x-axis and the annual land flux on the y-axis. And we have all these different model simulations that we can use to try to predict what this is going to look like into the future. And when we start using different models and different methods and different approaches, we understand that this is becoming way more complicated than it really was first thought to be. And of course this is complicated because this is kind of the system we're playing with. And I really, really enjoy this figure, and I really enjoy the complexity of this figure. We can think about the CO2. We have atmospheric CO2 moving through the stomata. It's interacting with these physiological machineries and the physiological gears driving photosynthesis. That's being converted to sugars, driving GPP and biomass. And then this is also interacting with all the forests and the forest products and land management strategies. But this also really highlights the underlying factors of soil water, as well as some of these macronutrients and soil fauna. And so all of these are causing bioperturbations and different things that are influencing all of these fluxes. And so I want to make this more complicated. There's another facet that's not being accounted for in time. When we think about how water use efficiency changes over time, we think about phenology, so we can think about circadian rhythms. We can think about phenology or other physiological seasonal drivers that are also influencing all of these dynamics that are also going to impact our use and the prediction of carbon and carbon sequestration over time. And all of this is interacting with the environment. And one of my favorite environmental drivers to think about is vapor pressure deficit. And so we have this really cool figure looking at how vapor pressure deficits are going to change into the future. And so this is work done by Darren and Kim Novick. And we can see we have generally VPD is increasing all across the United States. This increase in VPD is a really hot spot in the Midwest and kind of the Corn Belt area. But nonetheless, rising VPD is going to have significant consequences to photosynthesis as well as leaf canopy and carbon growth and carbon uptake over time. And so let's start with this. We'll just think about the theoretical limitations to photosynthesis based off of stomatal conductance. So just using some of the Rahm-Oren approaches to predicting what stomatal conductance on the y-axis versus vapor pressure deficit on the x-axis would look like. And the primary thing to take home is we have this nonlinear reduction of stomatal conductance as we increase in BPD. Consequently, decreasing conductance, we will decrease the CO2 input, CO2 integrating into the machinery, and then decreasing GPP. So this has cascading effects. But one thing that we don't really know, how does this change over time? And so we can ask certain questions as like, okay, is this relationship, is the slope of this line, is our reference or our intercepts the same as we move from springtime leaves to summertime leaves or peak growing season? And this becomes kind of important because we can think about early spring conditions. The environmental conditions are a little different to what they would expect, specifically being like there might be more frost events if we have earlier warming seasons inducing earlier leaf out, which would then put leaves at risk. Alternatively, we might have heat waves or early shock events that would disturb emerging leaf processes. Oh yeah. And also, can we do this in a fun way? So we have had some pretty cool events happen in the US over the last few years. And when we think about this, we have the solar eclipse. Hopefully some of you got to experience the total solar eclipse this year. But we also had the total solar eclipse in 2017. And we were able to use that solar eclipse to actually assess how some of these physiological processes occur at the leaf level. And so we can see this is the time series of photosynthesis during the 2017 solar eclipse. And we could see the predicted reduction in photosynthesis as solar radiation decreases. But we were able to scale this to the entire country and look at how the consequences of this are expanded. So what can we do with the 2024 eclipse? So this is results from the 2024 eclipse, and we just have the same kind of time series of the day, and then we have on the left figure, we have photon flux density or light quality, and we can see as we go, we're reaching some sort of maximum radiation, and then we get this really big drop, which is the solar eclipse moving across, and then we recover and progress throughout the rest of the day. But one cool thing that we are starting to see with solar eclipses is that the light concentration, you can see that the light quality and the cloudiness was really high, and then all of a sudden during the eclipse, the cloudiness dissipated and then returned. And so this is a really cool phenomenon that we're working on disentangling. Not the headline of this talk, but just another cool aspect. Alternatively, we had a nice midday depression of vapor pressure deficit. So we have vapor pressure deficit on the Y, we spiked it to the morning and then recovered. We have a gradient, so the gray to black is the profile across the tower, so from the ground to 30 meters. And the green represents the vapor pressure deficit at the leaf. So we can use that information to start understanding how do the plants respond during leaf out, and how sensitive are these stomatal and stomatal apertures to this fluctuation of midday vapor pressure deficit. And this is kind of important because all the information that we currently have resides at the peak growing season. So if we have this visual observation of leaf development, we have most of our physiological understanding occurs at the peak. And so we got to utilize this part of the leaf development curve to actually assess how does spring phenology and VPD interact and alter stomatal sensitivity. And the other great part was we got to do it from my backyard. And so we got to watch the solar eclipse. So similarly, but this time we're going to look at sap flux densities of developing leaves. So the top panel is a tulip poplar, while the lower panel is a sugar maple up in the Morgan Monroe State Forest. And so we can see we're increasing sap flow like we typically would. And then we get, as radiation and BPD drops, so does sap flux density. And then it recovers back to some midday. It's a really nice clean response in the sugar maple, and the tulip poplar is less clean, but we still see the same midday reduction. And so we can carry this on to looking just at the response in that window of what BPD and temperature dynamics are. And so we get this really nice nonlinear relationship as the theoretical model would predict. We also get this big hysteretic trends as BPD is cycling in the middle of the eclipse. But we really care is, are these more sensitive or less sensitive? And so we would assume that developing leaves would try to operate in the same fashion as mature leaves just to protect themselves from environmental degradation. But what we found was pretty cool so this was work done by Sandra Denham in the same forest. And so she published this in Tree Phys in 2021. And so I just compared her numbers to what we observed in the eclipse. And so we can see that there's like a 70 to 90% reduction in VPD sensitivity. And then when we think about this could be twofold. So as we are expanding leaves, we require a lot of pressure and a lot of energy to release that and develop the leaf. Alternatively, we also need potentially very, very high pressures to pull nutrients that we're constantly integrating into developing leaves. So this kind of makes sense. But what happens when the water runs out? This puts the leaves at a very, very dangerous situation. As soon as you have any water stress in the soil and you have developing leaves, what is going to happen? And so this allows us to carry on for what our next experiment would be. And so this is a kind of an example. So this is the 2012 drought, which was one of the most costly droughts in the Midwest and the Eastern Plains, causing approximately like $40 billion worth of damage. But we can see how fast this drought progressed. And so we have this top panel, and we can look at the drought monitoring signal. And in the Midwest, we see it's like, not too bad. But all of a sudden, by midsummer, this drought was really, really incredibly drastic and extreme levels across much of the Midwest. And so this was accompanied by both very, very little precipitation, reducing soil moisture levels, also by extreme heat and vapor pressure deficits. But one of the things that's hard to do is understand how these plants are going to respond. So we saw significant reductions in crops, but what happened in the forest is a much more challenging thing to disentangle. And the other concern was 2012 an anomaly? Most of the projections and most of the forecasts that we are actually seeing suggest that many of the eastern forests are getting wetter. Soil moisture is actually increasing. So that would suggest that we're going to get even more decoupling. We're going to get higher VPD in the same region that we showed in the Ficklin and Novick paper, but we're also getting more moisture. So that means we're going to have this decoupling of soil dynamics and atmosphere dynamics, which is going to induce a bunch of novel drought and stress scenarios. So thankfully, since we're using a theoretical relationship, we can expand this. Instead of using the stomatal conductance and VPD versus spring versus growing season, we can modulate this into looking at, okay, what happens when we add a 20% reduction of photosynthesis, or 20% reduction of soil moisture. And we can do this to an extreme range. And so then we can start asking different questions. Okay, so what is driving the primary reduction of conductance? Is it VPD or is it soil moisture? And we can look at this in a relationship to GSREF, or our reference or our intercept in this relationship. And we can just plot this in a theoretical perspective. So the primary take home of this would be we have a reduction of GSREF due to soil moisture, and the reduction of GS, or the relationship between peak and minimum conductance across this line. And we could see the primary takeaway is that we have, when soil moisture is high, VPD is the primary limiter. When soil moisture is very low, or soil stress is very high, VPD effects are very small. And so we have this interactive dynamic that's still challenging to predict. And partially is because, and so this moves on to, there's downstream consequences. So if we're reducing stomatal conductance, we kind of showed this earlier, we're getting less photosynthates, but this also influences transpiration. And so we can see we have this dynamic where we're decreasing transpiration, which would then also decrease carbon uptake, but we're also inducing water potential stress. And so the further negative we go down in this relationship of vapor pressure deficit to leaf water, potential would be a more stressful induced event. And so we know that both of these hydraulic and these hydraulic dynamics that are a consequence of the reduction of stomatal conductance are going to decrease carbon uptake while also increasing carbon cost and maintenance. But disentangling this in the field is a whole new level because we can't really control vapor pressure deficit, and we can't control moisture at the same degree that we can in controlled scenarios. And especially if we want to ask this kind of question, what to extent are VPD and soil water potential independently and interactively influencing gas exchange, water potential, biomass accumulation, and growth? And does this vary when we think about water use behaviors? We have some trees that are very drought tolerant, and we have some trees that are very drought intolerant. And so to really do this, we have to use a growth chamber experiment to actually assess and control these kind of scenarios. So this is our growth chamber work that we've been doing, trying to disentangle this relationship. And this is really beneficial because we can look at the vapor pressure deficit. We can control it across different times. We have a control group where we have a nice low VPD level. There should be bars in here. Nonetheless, and there's two different phases of vapor pressure deficit that we induced. So you can see we have this nice step function, and then the step function increases. So we actually moved from approximately 3 kilopascals to 4 kilopascals throughout the progression of this drought. And we also used a pulse-based soil water potential approach where we would let them drop and then repulse them with a small precipitation event, trying to emulate an actual scenario that we would see in the field. And so from this, we have multiple species, or 2 species, nested inside here, so we can actually start assessing how different species and how these different species would change the forest and forest function over time. So this is just the fundamental associations between isohydric species, or much more water conservative species, and we're going to leverage our sugar maples to represent that cohort. And then we have our more anisohydric, or our liberal species, who will allow their water potentials to drop in order to keep stomata open to maintain photosynthesis. And so we can see that we have this spectrum of relatively drought tolerant to drought sensitive, and we can assess different traits. So we can think about how water potential is going to be regulated. So sugar maples are expected to close their stomata, and while oak species are more predicted to maintain open stomata, and this will alter leaf content and how these leaves can handle it, as well as wood density and biomass accumulation. And if we kind of forecast this into the future, we could assume, okay, if we have a selection for one of these species, this is going to alter a much larger ecophysiological perspective or an ecosystem scale perspective. So if we look at the actual data, we can start teasing apart, okay, so this is a normalized assimilation rate versus the days of drought, and there also should be a color indicator in here that descripts or deciphers the 3.5, the 3 to 4 degree BPD levels, but for some reason they're not showing up. But nonetheless, we can see that the drought effects seem to take a while before we see any real treatment separation. And so we have sugar maple on the right, and then the anisohydric oaks on the left. And we can see that this trend follows both assimilation rates or photosynthetic rates, as well as stomatal conductance. And so, but the main thing that we noticed is that, okay, none of these, and these were little sapling trees in the greenhouse or in the growth chamber, none of these separations or effects were really noticeable within the first 30 days, which was kind of surprising. We assumed that these saplings would be relatively stressed within the first 30 days, even under high VPD. But we also noticed that the VPD condition was either higher, especially for the oak species, or not that much different than the control species as we observed in this, suggesting that the plants can adapt above ground biomass or above ground leaf physiology to account for higher BPD, while the below ground limitations or any drought scenario that has soil moisture plus VPD drought is a much more detrimental impact on leaf level physiology. And that's kind of a hard picture to take apart, and so that doesn't really answer the question of how interactive or independent these different drought scenarios are. So here we actually took a different approach, thinking about the days of drought on the y-axis again, but the limitation to photosynthesis or the limitation to stomatal conductance. And the top panel would be photosynthesis. And so what we have here is the different treatment colors are the same, where gray is soil moisture, black is VPD, and red is the combined. But we also just essentially took our cumulative effect by adding the VPD effect plus the soil moisture effect, compared that to the empirical observations. And we find a kind of interesting dynamic when we actually look at it this way, where we see that the anisohydric oaks, they have very little limitation to both photosynthesis or stomatal conductance. And actually, VPD enhances photosynthesis in the white oaks, while we see the sensitivity, especially latter parts of the drought, are really high in the white oaks. But another key takeaway is that when we take this cumulative approach, we see that it is nowhere near, typically nowhere near, the actual empirical combination of soil moisture and vapor pressure deficit limitations. And so, okay, so this is kind of more resembling of what we would expect from our isohydric anisohydric paradigm. But how does this translate to actual biomass accumulation and growth? And so we also, at the same time, we were measuring the cumulative stem diameter and cumulative stem growth throughout this entire experiment. And interestingly, approximately when we crank the VPD to 3 to 4 kilopascals, we can see that all except for one cohort reached some sort of threshold. And additionally, we also started to see that these treatments started to separate, but not exactly in the same way that we saw with the gas exchange. So nonetheless, we had high levels of growth in the control and soil moisture, but the oaks did very well in this situation. So even though the the control oaks did very well. All the other oaks in the biomass accumulation was very low. And we can look at this in a different way, and we can look at this in an organ-specific manner. And so this is an effect size, so we can assume this is a Hedges-G's effect size, so the more negative, the more detrimental the effect, the more positive, the more enhanced the effect would be. And if we break this down into total biomass, stem growth, root biomass, and leaf biomass, we could see that the anisohydric oaks, the ones who are keeping their stomata open, are being the most impacted. So that would suggest that they might be able to do more photosynthesis, but they're also losing more carbon or not growing nearly as well. So this poses an interesting question. While the isohydric sugar maples, who showed really high sensitivity and high limitations to photosynthesis and gas exchange, were impacting the growth and growth partitioning. So what we really are taking home is that gas exchange is not mirroring biomass accumulation. So this is an interesting component. So what would potentially be causing this? And how can we decipher some of these results? And so, yes, once again, the limitations in oaks were more pronounced in the biomass accumulations. And so one of the ideas is, okay, maybe these trees are using carbon differently. And so we have maybe an upregulation of carbon maintenance. And we think about this from a different perspective. And so while you might be able to maintain higher photosynthetic rates, you might have to maintain those photosynthetic rates to maintain the same level of respiration to get through the drought. And so what we have here, so this is continuous leaf water potential measurements derived from a stem psychrometer. And so this is our control group where we have every week or so we averaged everybody, and then we measured the average soil and water potential in that cohort. And we can kind of see how the treatment effects vary. Oh, I didn't label these well. But this would be your high VPD, or no, this would be high VPD, no soil moisture. This would be low soil moisture, regular VPD, and this would be the combined effect once again. And so one of the ways we can actually disentangle this, of course, as we drought the plants, the water potential drops. This is kind of well known. This is some of the basic physiology of how water moves from the roots into the plant, into the leaves, to the atmosphere. But one thing that we can decipher from this data is we can start understanding stem pressure stabilization. And why this is important is thinking about when we have like a resting heart rate versus an active heart rate. And so if your body doesn't go into a resting heart rate position, your body doesn't have a normal recovery at night. And so plants kind of can do the same thing. So if we think about what a normal water potential dynamic could look like over time, where we have this flat peak, where we drop our water potential, and then we recover, and then we hit some recharging peak again. If we look at unstabilized pressures, we would assume that there's this continuous roller coaster where we're moving from a increasing, dropping, and trying to increase, but as soon as light and VPD reemerges, pressures are going to automatically drop, trying to account for transpiration and stomatal conductance. And so we can use this, and we can calculate this slope of the water potential every night and try to understand how stabilization is impacted across these different treatments. And with different treatments and without maintaining pressure stabilization, you're not able to divide your cells. And so when we think about this, if we look at the hydraulic stabilization of leaves at night, so this is just the nocturnal drivers, we have our different treatment cohorts, and this is our nightly slopes of those relationships. And we can see the controls cohort typically is well recovered and is almost always below zero. Alternatively, as we induce soil-induced droughts, there is a problem where we're not recovering or we're not recovering to a stabilized or a relaxed state. But the biggest concern is when we're seeing this increase of vapor pressure deficit. And so when we see an increase of vapor pressure deficit in the day, you're dropping the water potential and you're having a hard time recovering this pressure. And so we see even with not soil moisture droughts, we're having this complex dynamic that is disturbing nocturnal water pressures. And while this might be, okay, that's not very cool, nocturnal water pressures are not that great, but we're learning a lot more about nocturnal physiology and especially how important it is to regulating plant growth. And when we think about this from a perspective of when do plants grow, when do trees grow, when does the wood get laid onto the xylem, we start to see this paper, this Zweifel paper that came out, showing that the contribution to growth on an average basis is really at nighttime. So we have this really peak time where we're actually not stabilizing water potentials, meaning that cells can't actually divide, cells can't actually maintain turgor pressure. So we're disrupting the same period, which would then also reduce any growth, which would then also increase any level of cellular respiration or maintenance respiration at the plant level. And so, yes, without any of that, we can't maintain pressure. We're accelerating our respiration rate. And one of the problems when we do studies like this is that it's really challenging to measure plant level respiration of all these different organs at continuous rates. And so for this kind of part of the talk, it's just really thinking about how do these VPD and soil moisture stresses impose novel or reductions of growth and recovery. We also have these limitations that don't mirror each other. So if we're only measuring gas exchange and trying to forecast biomass accumulation, we're going to start understanding that there's maybe bigger problems in our carbon balance and our carbon approaches for how we're assessing the carbon productivity might be skewed depending on water use strategies and how these leaves behave under different drought scenarios. And just a reminder, as expected, the species behaved, when we looked at gas exchange, the isohydric species behaved just like we would expect. And much of that is because a lot of our fundamental understanding of isohydry and anisotropy is built off of the dynamics of gas exchange and leaf level physiology during the day. But we're missing a lot of things without these continuous measurements. And so we're starting to get better at this as we develop more reliable and more robust tools to measure continuous water potentials, specifically at night. And so, yeah, we saw the biggest impacts on the trees that we thought were going to be the most robust and most drought tolerant, but they actually showed the most reduction in growth. And so with that, I also want to think about some of the so we really focused on abiotic perturbations so far in this talk, thinking about VPD and some of these like circadian dynamics. So we can also spin these into some of the biotic perturbations, like when we talked about that dynamic of earthworms and all these perturbations in the soil. We can also do this, and we can also leverage another pretty cool charismatic event. So when I first got to Indiana, everyone was anticipating this crazy emergence of billions and billions of cicadas. So these are periodical cicadas, which are pretty cool insects, and they're actually root parasites, and so they burrow into the ground and they live on the roots, and they suck root sap for 17 years, and then they all emerge at once. And so all of these cicadas are coming out, and there's 17 different broods. So we can see that somewhere in the eastern US, one of these broods is emerging almost every year because we have a mixture of 17 and 13-year-old broods. And so somewhere, like this year, we had a really big population. Both of them popped in this area right here this year. And so there was a big spectacle of two broods coming out in the same city in the same time. But how does these insects, or how do these bioperturbation events affect the actual ecosystem? So we know from some of the work being done at Indiana University that as these cicadas emerge, each cicada, billions of them, emerge from their own hole. And as these cicadas emerge, they pit the ground, and some of these pits can go really deep, like a meter or so below the ground. And so we had some really cool research looking at how these influence hydrological flows, and how does these cicada burrows alter soil moisture and soil water infiltration. And so we can see there's a big increase in the actual hydraulic saturated conductivity, so we increase the ability for water to be sucked into the soil between disturbed and or soils containing burrows versus no burrows. And if we average this out, it can be up towards 80%. But on a carbon or a more biogeochemical perspective, what would this look like? So if we have millions of these burrows throughout the forest, and we have this channel that's a direct channel between the surface of the atmosphere all the way to the root, we can think about infiltration, and that would benefit some of the root and some of the root physiology. It could also provide an anaerobic condition. So these anaerobic conditions would drive out oxygen, producing reductions in soil respiration as microbes and everybody were being drowned out. But there's an alternative hypothesis where we actually are pulsing the deeper channels with oxygen and oxygen diffusion throughout the soils. And then so that would also increase the amount of percolation. So we have all this dissolved organic carbon and dissolved organic nitrogen percolating deeper into the soils, which would stimulate microbial function and root uptake, which would then stimulate soil respiration. And so we wanted to test this in our forest, thinking about, okay, so we have different microbial cohorts. We can separate this into arbuscular mycorrhizae species and species that leverage, that typically have a lower leaf turnover or more sensitivity to nitrogen versus the AM species who are less sensitive to nitrogen. And we could think about this in a waterlogged scenario where we would expect both species, regardless of microbes, to be decreased as number of holes increase. So more percolation, decreased CO2 flux. Alternatively, under aerated soils, we could potentially get this interaction where AM trees are less sensitive, but ECM trees are more sensitive. And so we wanted to ask, does this influence soil respiration, and does this influence, or is this influence microbially specific? And so when we did this study, one of the hard parts is you have to wait, because we don't know where the cicadas are coming. We know there are going to be lots of them, but we want to do this on a very specific name. So we waited, and they started to emerge, and then they emerged, and there were cicadas everywhere. They were not underestimating the billions of cicadas. And I mean everywhere, covering my car, covering us in the field, covering my dog. They were getting stuck in our gas analyzers constantly. And so this was posed to be a problem. So once they finally emerged, we were able to locate the holes. And so we used essentially a paired collar approach where we could find areas right next to the trees with a paired collar or with a hole and no hole, at least one hole. And while this is quite a messy relationship, we still get some decent separation, especially as we get to warmer conditions. And so we have soil temperature here on the x-axis, and we have our soil respiration dynamic on the y-axis. And just from our paired approach, we can see that holes versus no holes, there is an increase in this relationship. While it's not very huge, it's a much easier perspective if we think about it from what is the sensitivity. So we talked about stomatal sensitivity. Now we can think about sensitivity to respiration or temperature respiration to temperature. So this would be more of the biochemical Q10 dynamic that we're associating with enzymatic function and microbial rates, and microbial respiration rates and so forth. But if we actually use this relationship of the Q10 dynamic between regions with holes and without holes, we see that there's pretty decent, like approximately a 30% increase in sensitivity to soil respiration. And so this was kind of interesting. So we can say, okay, if we have more cicadas, warmer temperatures are going to drive more soil respiration in these regions. But is this microbially driven, or is this just based off of the hole density itself? And so once again, we were able to we can look at this from a species level or a functional type level where we separate these into our AM trees or our ECM trees. And interestingly, we can see that we had the opposite of our initial hypothesis, thinking that the ECM trees, who are more nitrogen limited typically and have lower decomposition rates of their leaves, showed essentially no change over the course of the different hole densities, while we saw a much greater response between the AM trees. And so while this is also kind of interesting, but what does this really mean at the ecosystem scale? I took one collar, that's approximately 8 inches, and measured that at that surface. If we actually scale that to a a 1 square meter, that would equate to approximately 200 cicada holes per square meter. That density was not observed in the forest. So it's hard for us to translate this directly from our soil respiration chamber measurements to our ecosystem scale. But we have been very fortunate to be in Indiana, and we're very fortunate to have a long-running flux tower. And so we can address this in a very similar way using our eddy covariance approaches. So thankfully, this was our 21st year of running the AmeriFlux Tower, so we had a little party. And so conveniently, there was 2 cicada emergence events that occurred within that 20-year window or 21-year span. And so we had 2021, of course, which was a pretty cool year, and then the 2024 year, which was also a cicada emergence event. And then if we compare that to what it looks like during the average, we can see that ecosystem scale Q10, or that same dynamic of the temperature sensitivity of respiration, we can actually see is significantly lower than any of the cicada emergence years by 20 to 30%. And so the primary understanding is like we know that these cicadas might only emerge a couple times a year or one time a year in certain locations, but they can drive a significant increase in CO2 respiration. And it's likely driven by increasing microbial sensitivity to temperature and allowing dissolved organic nitrogen and dissolved organic carbon to penetrate deeper into the soil, activating more microbes and activating different layers of the soil. And then, so we also have that these cicadas are playing a really important role in carbon cycling, and because they're beyond just the carcasses that are being integrated into the soil, we can see that this is also just a pulse vent as the cicadas even emerge. And the other thing is this might actually be an underestimate of the effect, because when we think about, we had to wait to find the holes. The greatest effect was probably immediately after they emerged, because then you were releasing all the CO2 that was bubbled up in their chamber as they were excavating the CO2 throughout the entire profile. And so with that, I'm approaching the end, but we're looking at, I just want to remind that we can leverage really cool events to understand physiological and ecophysiological processes. So far I've been able to use 2 solar eclipses and a cicada emergence event. And so we can actually see that there's really interesting dynamics occurring from these events. And then additionally, we have a lot to still disentangle between the VPD dynamic and soil moisture dynamic that is impacting eastern forests. Even though the drought effects of the eastern forests seem less severe, less grave, compared to what we see in the visual representation of what we experience in the western US, some of these impacts on actual carbon storage and carbon sequestration may be overestimated or underestimated based off of the VPD conditions that we are understanding. And then lastly, the biotic perturbations on CO2 effects and CO2 storage in the ecosystems is becoming more and more important as we think about where the CO2 and the fate of these different processes before we start increasing more of the free CO2 capture programs. Maybe we should study more of our native capture programs. So with that, thank you guys, and I'll gladly field any questions. Go ahead.
Thank you. I am a soil in particular, so the cicada emergence is really interesting. And I was just wondering, do you have any thoughts on the mechanism of this AM, EM? I mean, I know that there's likely different communities below there, but do you think there's any other structural differences or anything that could be driving the effects there?
So the primary question is thinking about what alternative mechanisms would be driving the differences in CO2 evolution between AM and ECM cohorts. Yeah, I mean, there's a lot still going on there. So there are some caveats to thinking about this. One is this is a very diverse forest. If this was two different, if we had like a nice sugar maple stand versus a oak stand where we could actually disentangle and be confident that I was not measuring oak roots underneath those chambers, that would be one advantage, but we couldn't do that. And we also kind of know that most of the fungal biomass is probably not under the primary bowl, and it's actually has a greater dissipation as it reaches beyond the primary bowl of the tree. So there are some limitations, of course. But yeah, I'm not exactly sure the other mechanisms that would be influencing this, other than like the nitrogen and the turnover rate, thinking about the decomposition of home leaves versus intraspecific leaves. Go ahead.
You introduced time as a complexity early on, and years ago, ecophysiologists were fixated on thinking about whether leaves were capable of exploiting sunflex, for instance.
Sure.
We knew that light intensities during the sunflex were way above a traditional light saturation measurement, and I think it was mostly Bob Piercy's work that showed that the photosystem was capable of absorbing those quantum and then storing them and then using them more efficiently. So I was wondering about vapor pressure deficit and diurnal variation. I mean, how much CO2 enters the leaf during periods when it's not showing up in gas exchange in a photosynthesis measurement? I mean, is there a way of disentangling that? Is there much diurnal variation in BPD that can be exploited? Nighttime, for instance.
Sure. Yeah, I mean, absolutely. Like nighttime BPD is a really interesting, complex question.
Can you repeat the question?
Oh yeah, sorry. So the question is essentially, what is the effect of like nocturnal, or the relationship between CO2 immersion into the leaf relative to VPD concentrations in a general sense? That's a really cool question, thinking about, and a lot of what we know as ecophysiologists, though, has been fully developed during the day. And so out of convenience and out of magnitude, like we didn't really have good tools to measure really small fluxes. And we know some of our tools are highly uncertain. And so, but we're getting better and better at this, and we're also realizing how important like nighttime is. And so nighttime BPD, I think we're just entering into a new era of understanding how BPD is also influencing physiology at night. And which is actually going to be more important, I think, because we're starting to learn that growth is at night. And so I don't think we have a very good grasp of how the coordination between CO2 uptake and nocturnal BPD is. But we do see a lot of transpiration occurring, which would suggest that there has to be diffusion the other way. Whether that's, I mean, yeah, so if nighttime transpiration is occurring, there should be CO2 diffusion occurring. That's just, was it?
I was really kind of like blown away by the nighttime growth results.
Sure.
Of importance of growth.
Yeah, absolutely.
So cool. How does that vary with latitude, right? So I'm thinking about high latitude systems where it doesn't get dark in the summertime.
Oh, that's a cool question.
Not quite as much. Do you think that varies with latitude?
I would say yes. But that would be ripe for a proposal if we put some continuous dendrometers on your trees up in Alaska. That would be really cool.
Can you repeat the question for the virtual audience, please?
Oh, yeah. So the question is thinking about how regions or latitudinal dynamics that don't receive the nice diurnal gradients of nighttime and darktime dynamics influence stem growth, essentially. But yeah, I think we're just learning about this right now. And this goes back to, as ecophysiologists in general, we didn't have really great tools. And now we're getting much better tools to measure really, really small fluxes, and even fluxes such as like the stem oscillation. And so that's how we can tease apart how the stem growth is changing. And so I think we're just entering into as Raspberry Pis and all these little tiny microcontrollers are becoming more and more prevalent. We're just entering into a new phase of new ways to measure this. So I think that would be a cool approach. I would be interested in digging deeper in that.
We have a question from the virtual audience as well.
Okay.
Clive asked, how long does a cicada hole persist?
Oh, okay, that's fair. So cicada emergence holes like varied. So there was a consequence of, one, are they in a collar? But typically they'll last for like 6 to 8 weeks, so several months. And so, yep.
How do nutrients modulate like tree hydraulic functioning and resilience to drought?
So how do nutrients modulate tree function to drought? Well, I think there's that can go in different ways. One, what is the drought? So if it's soil moisture drought, we assume that depending on the stage and how it's going to be impacting, if it's an extreme drought, we are going to actually disassociate roots from the soil, so then you're not going to be able to take up nutrients very well. But if it's a VPD-induced drought, you actually might be able to increase your nutrient uptake at a faster pace. So like, for example, we saw oaks typically had higher stomatal conductance and higher gas exchange under VPD, elevated VPD. And that might be consequence because they're able to readjust some of the leaf mass per unit area or some of the leaf structure or the leaf anatomy to account for that vapor pressure. And so they might actually be able to increase nutrient acquisition and counteract the drought effects, essentially. Yep.
Thank you for your talk. I was wondering if you know more about the relative abundance of these anisophytry and isophytry species in the forest that you study, and if you can make like a guess of what will happen with the full stand growth in the forest that you study.
Sure. Yeah. So the question is like, what is the relative abundance of isohydric versus anisohydric species, and how would we potentially forecast that into the future? That is a good question. So a lot of what we know about isohydry and anisohydry is done at the leaf level, and so, but we do know that this isohydric to anisohydric dynamic is a spectrum, and we're also learning that individual trees and individual plants can switch. So like there's a really cool paper from Jessica Guo looking at, I think it's creosote, the desert shrub in Arizona, creosote. But you can see that the monsoon alters its water use behavior. So during the dry season, it'll be incredibly isohydric and be very, very, very conservative. As soon as the monsoon hits, it's liberal as can be with water because it's just like, I'm taking up everything, all my nutrients are coming up now. And so it's hard to say what would the forest look like because we don't have that good idea of how flexible is an individual. We know on a general perspective that tulip poplars are very tightly regulated. They're even drought deciduous, so they'll drop their leaves. So I can tell you, if it got really dry over long terms, they would not do well. And the oaks don't drop their leaves. And so we can make really large general consensus predictions, but we don't know enough about the entire spectrum of diversity to make really comprehensive predictions, I don't think. But that's a good question.
We have another question from the virtual audience. Miriam Johnston said, thank you. What's your next big event?
Well, there's an eclipse in Spain in 2026. That's been talked about. I don't know yet. That would be, yeah. Yep.
So the cicadas are spending either 13 years or 17 years feeding by the thousands on these individual trees underground.
Yes.
And the other thing that happens just before they build their tunnels and emerge, allowing water infiltration and changing the microbial dynamics, is they're not feeding anymore.
Yes.
So how would you distinguish the impact on trees of the sudden cessation of this underground herbivory from the infiltration of water affecting the microbial communities below ground?
That's a really cool question. Yeah, so the cicada life cycle is very
Can you
interesting.
repeat the question?
Oh, yeah, sorry. So essentially, the question is thinking about the trade-offs between being parasitized for 17 years and then being released, and how would you separate that effect from the carbon effect from the emissions? Yeah, that's a really cool. I mean, so that was one of my original ideas, was to like, okay, we're going to instrument all these trees. We have a couple years of data. Can we actually see like a parasitic release? All these trees, are they going to just start cranking out CO2? And that might be what's happening in the nighttime respiration at the ecosystem scale. We don't really know if it's microbially derived CO2 emissions or the trees just growing really, really rapidly. And so that's hard to disentangle. Maybe we could use some like isotopic signatures if we had something cool like that on our tower. But like from an anecdotal perspective, it was a pretty amazing year on how many animals. And so like the productivity of the forest was immense. Like I've never seen so many rattlesnakes in my life. But like there were so many fat, happy rattlesnakes because they're so satiated from the cicadas. So I'm assuming that the trees also had some sort of release. But the other aspect of the cicada life cycle is like, okay, so at the same year you're being released, or maybe a year before as they traverse from the roots to the surface, the cicadas are doing pretty significant damage to the lateral part of the branches. And it's like where the female cicada will drill and oviposit into the lateral branches of the tree. And so this actually decreases the photosynthetic carbon uptake. So there might be an offset where you might gain more carbon, but you're going to lose it right away as you invested in that leaf area and dropped it. But that's a cool question.
Any more questions for Dr. Beverly? Okay, I suppose that's time. Thank you all very much for coming, and we'll see you at lunch.
Right.